Yeah, today we will talk about a study we published on Nature Plants about the attractivity of plants on scientists and research community in general. And the idea was born during the Italian lockdown, when I was writing the introduction of this article about Tephroseris barbisiana, which is a not so beautiful plants from southwest Alps. And this area is at the boundary between France and Italy. It is an hotspot of biodiversity, one of the Mediterranean hotspot biodiversity, and it is a very big center of diversification of plants. So in this area, which is the area of the Mercantour National Park, more or less, we can find more than 100 endemic species, 78 rare species, and this core of or particular species, I think that they characterize the flora from this area and make it unique and particular. And it is really interesting to study it from the taxonomical, biogeographical, conservation point of view, and also evolutionary stuffs are really interesting to make it on this particular kind of flora. And so I started from the idea that this flora is rich of particular species, but when I was writing the introduction about my endemic plants, I didn't find nothing published on a scientific journal about this plant. And I think that why there are a lot of articles about few very nice and beautiful plants, and no articles were published about this ugly plant. So starting from this idea, I imagined how to make a publication about this point, because I said, yeah, it seemed true, but probably if we apply some statistics on this idea, we can really publish something about this bias of aesthetic in botanist head. And so I also focused on this flora to make this work, because maritime Alps flora is studied extensively for over 2 centuries. So the number of taxon is really stable since 50 years, more or less. We have a quite narrow range of plants, so we don't have really confounding factor, including very different biomes. This is the case, for example, if we compare alpine plants with plants from savanna, probably data are really skewed by these really different biomes. And this area is mostly studied from researchers from France, Italy, and Swiss. And so we don't have big cultural biases in the perception of the reality. In order to understand which is the research interest about a specific plant, I think that the proxy obviously is to study directly the scientific publication. And France, Italy, and Swiss are between the top 50 countries published more in the last 50 years. And we have published in the 2016, we reached the 2 million of published papers. So published papers is a very, very big database to study. So I think that study scientific publication is a very good proxy to interpret research interest. And in fact, there is a rising discipline which called bibliometrics, which use statistical methods to analyze scientific publication per se. So you directly study how research published on the different journals, on the different topics. And we used to extract the name of the plants that we were interested in directly from the scientific publications. There was a little problem in this because everybody knows that each plant has an accepted name, but we have also synonyms, unresolved names, or unplaced names. And we use the plant list database, which is the official database to know if a name of a plant is a synonym or a accepted one. And we discovered that there are a very big number of synonyms and other kind of names to make to have an idea if we consider that probably in the world exists 3082 number of plant species. In this database, we have more than 1 million different names. So we restrict our analysis to 113 target species for which we were sure about the accepted name and synonyms they had. So we exclude, for example, unresolved species or subspecies which are really difficult to manage with. Then, starting from the long list of species names, we searched for it in the title, the abstract, and the keywords of all the articles included in the Web of Science database. So in the Web of Science database included all the scientific papers between 1975 and 2020. And then we had an output of 280 papers, which are not so much, but we have to consider that 43% of the species had no published papers. This is also the case of the Froseris balbisiana, the species which I studied. Then, after the extraction of the number of the research paper for each plant, we characterized each plant species using two main questions to inspire the traits we go to extract, which are: why are we interested in Southwest Alp species? And we are interested in the species because Southwest Alp are a glacial refuge, because the species are rare or endemic species, because some of these species are really peculiar taxonomic position, because they live in unique environments, or this is just some examples. And moreover, we was in the lockdown, so we can't go out in the lab or in the fieldwork. So we used only traits that were available at home. And we extract data from a lot of different databases, such as the French Tela Botanica, or the Italian version of Tela Botanica, which is Acta Plantarum, Flora Helvetica, Flora Vegetativa, and many other different databases were used to extract data. And to make just a fast screen, a quick screen, we extracted data from taxonomy, the plant life form, the flower colors, the conservation status of these plants. We characterized also a little bit the ecology of the species using Landolt index values, then stem size, flower diameter, flowering duration, altitude data, the distribution of these plants thanks to the GBIF database, the congeneric species, and the research interest, as I already explained. And then we put all these data in a model. We use the generalized linear mixed model, the GLMM, which is an extension of the GLM, the traditional GLM, which use linear prediction with a random effect, so with a bigger ability to explain the variability of the data in effect. And also we can use non-normal distribution. In fact, we use the family of as the taxonomic family of the species as random effect, and where we use not a normal distribution but a Poisson distribution. In fact, we had counts and not normal data, so it was at least mandatory to use a Poisson distribution. In the modern generation, we also analyzed that not only the number of scientific papers published for each of our 113 species, but also we tried to use as a response variable the number of citations and the H index of the papers. But we discovered that the correlation between the three factors was really strong, so we focused on number of papers, which is really easy to understand, to explain. Then this is a very graphical abstract of our experimental design. So starting from the endemic flora, we extracted 113 species, and for each of these species we extracted the number of papers from the Web of Science referred to the species. And for each of these one species, we extracted ecological factors, phenotype factors, and rarity factors. And the ecology group was composed of elevation range, maximum elevation, and the PCA able to make a reduction of the land health indication values. Then there's some traits such as flower color, flower size, stem height, and flowering duration. And lastly, the rarity of the species, the range size or the dimension of the area of each of the species, the extinction risks categorized with the UCN categories, and the taxonomic uniqueness, or otherwise the number of congenerics. Here you have the photo of Berardia lanuginosa, which is an example of the peculiarity of this flora from Southwest Alps. This is the unique species of this genera. It is endemic of the area, so its range size is reduced. Is a very small plants. They live at very high altitude. This plant is really well studied, and because it has a number of different point of interest regarding also the traits we extracted for. This is what a little bit the idea we followed to select the factors to study. And then we started to apply the models. And for the first analysis with various partition analysis, we discovered that ecology and rarity explain nothing or very few of the variability of the number of papers. And almost all the variability explained in our model was centered in the phenotype. and also the random factor, or otherwise the family of the species, explained a big part of the variability of our model. But what is really interesting is that phenotype is the bigger proxy in our model explanation. Then, from our results, we discovered that three different factors in particular were significantly correlated with the number of species, which was the color of the flower. So blue-colored flowers significantly more studied than all the other flowers, which is followed by white, and also red, pink, and yellow flowers tend to be more studied than the baseline of the green-brown flowers. A second result is the negative effect of flower size on research interest, where we found that greater stem height imply that species are more conspicuous or taller. So there is an inflorescence effect on the research interest, probably species taller in dimension of with bigger inflorescence are preferred by botanists to be studied. And finally, we found a positive effect of range size on research interest. This is a weak effect, but it could be correlated with the number of research groups who can access the area where the plants grow. If the area is very small, is only maybe a little area in France, is only some few groups from France are interested in the species. But if the species is endemic of the whole West Alps, can be of interest for French, Italian, or Swiss research groups. Then, to conclude, what I found really interesting to discover in this model is what we didn't find. That is that we did not find a correlation between extinction risk of the plants and the number of publications. So we can probably trace the presence of an aesthetic bias also in plants. This is already discovered for animals, but probably also botanists are influenced in their choices by aesthetics of the plants. And after the publication of this paper, many people asked me some question about these findings, and I started some reflection of probably there is also some bias introduced in how funds are assigned to researchers and how science interface with the conservation stakeholders. So this is just few questions that I think that everybody we have to start to reflect. And to conclude, I want to thanks to all the teams, guys, it was an absolutely informal teams, and Matteo Chialva, which is connected, I saw, Jacopo Calevo from Naples, Filippo Bertoni from Berlin, Kingsley Dixon from Perth, Australia, and Stefano Mammola from the Italian CNR and from ESCI. And so I stop here my presentation, and please ask me what you want. Thank you. I probably don't hurt. You have the mic muted.
Yeah, sorry, excuse me. So thank you very much. It was very clear, and this is an awesome way to do science during lockdown. You have done everything during the first lockdown in.
Yeah. During I think I started this work in April 2020, and so I had a lot of time to think and to do these kind of things.
All right. So does anyone has question? Otherwise, I have other questions. So normally you're all able to activate your microphone. Okay, so I have a first question. Would you have enough data to test this trend, through time? Because is this trend or do you have a feeling, or can you test it if this trend is stable or is it worse? Are we always publishing more through time on these flashy plants? Or do we diversify more?
No, we tested it just once, but I think that it could be interesting to test it also on another flora. I don't know, on the UK flora, which classically is really well studied, and but we never had time to test it, and probably the blue has some trace with also the blue color of the plants maybe have some trace with the geographical position. So these are things that we have to test.
Yeah, sure. Yeah, about this blue color, I'm not a specialist of plants and neither of this flora in the Alps. And so in classical flora books from the Alps, are there more blue species described?
Yeah, on the Alps there are several families which are characterized by the blue color of the flower, such as Gentianaceae, which is one of the, or Alcinaceae. And but we used to put the family as a random factor to delete these
This effect.
kind of effect. Yeah.
All right. Does anyone else has a question? Okay, otherwise I've got another one. Sorry, what was the number again of species that you finally kept?
113. 13.
And sorry, I didn't get. What is the repartition or the distribution? I mean, of all this of the abundance of citation of all these species. Was it okay? Do you have one or 10 major species always described versus just one publication with one species? This is my first question. And then, how did you deal? Did you select also publication talking about several species?
Okay. Yeah, the distribution was composed with 43% of zeros, which is surprising that the bigger number was 1, 2 number of publication, and that there were few species with more than 5 publications. but only one was really high in number, was cut out from.
Okay, it was an outlier. Okay.
And the second question.
How did you deal when you? In fact, it was text mining, I guess. How did you do? You extract all the abstracts from PubMed, and then you kept the publication with 2 or 5 species, and it's very technical.
The problem in this approach is that to use the publication as a valid number in the analysis was to have the name of the species, the title, the abstract, or the keywords. And so we excluded a lot of ecological studies, because in ecological studies, plants are just maybe in a table in the results, and there is no bibliometric tool to extract the data from the tables in the results. And usually the more common tools used in bibliometrics just extract from the topic, so the title, the abstract, and the keywords. And yes, in some keywords or abstract are listed all the species included in the paper, but usually are taxonomical taxonomy papers.
All right, and okay, it's very technical again, but so it was a lot of you automized. I mean, you coded to extract all the data, but then you have to do a lot of checking also. And you were talking about using and applying this method again to other flora. So did you? In fact, I didn't check in your article, but do you give the code if other people want to do it? This is my first question, and second, another question. How many times did you spend to do all this manual checking?
Many times.
Okay, a lot of nights. Okay.
No, the code to extract the bibliometric data, I don't remember if it is included in the R script, but there is an R script that you can download from a Figshare, and you find the DOI, DOI from the article.
Yeah.
And I checked everything manually, more or less. Also because 2080 papers are not so much, but I didn't find errors.
Okay, great. Roland?
Yes, I have a question. I was wondering if this sort of, can you hear me?
Yes.
Okay. No, I was wondering if this sort of study could be extended to other databases like herbaria. Because, for instance, herbaria are used to infer the distribution of plants or and variations in distribution of plants. And you could imagine that if botanists tended to oversample some species, you could get biases in their distributions, and you could underestimate, for instance, the distribution of some plants if they were under-collected. I don't know if this is the right term. So do you think it's possible to extend this sort of study to herbaria using databases by mining automatically herbaria databases?
Yeah, in fact, I think that most of the major European, at least, herbaria are included on the GBIF database. which directly associate the herbarium specimen to a geographical point. So I think that it's possible to do this kind of analysis, and I don't exclude that something is already published on it.
All right. Oh, wait, I might shut down one more mic. Okay, it's all right. So, does anyone has a last question? Okay, and so just finally, you keep on working with microbes. You don't work anymore with plants?
No, I work now. I'm working mostly on microbes, but I'm also working on genetic population of rye, the Secarace reale, and so but I don't know. Probably I prefer plants than microbes, but it's really hard to work just on plants because I have a background which is really strong on microbial ecology. So it's also a shame to lose everything.
All right, perfect. So thanks a lot. But all these methods are, yeah, they are very applied with microbes. So yeah, it's nice to have done it with this kind of data set. Very nice study. So people can contact you, and if they have more questions, and I wish to thank you again for giving this first seminar. Yeah, from the season. Wishing you the best.
Thank you very much.
Thank you. Bye.
Have a nice day. Bye.
Bye, Martino. Thank you.